Perfect. So let me introduce, last but certainly not least, invited speaker today, Professor Vida Jamali. She is an assistant professor and Daniel B. Maury Faculty Fellow in the School of Chemical and Biomedical Engineering at Georgia Tech since August of 2022. Before that, she was a postdoctoral researcher at UC Berkeley, working with Professor Paul Alvasados in the College of Chemistry and Kavli Energy Nanoscience Institute. And she received her PhD in Chemical and Biomolecular Engineering from Rice University in 2017 with Matteo Pasquale. And her research group at Georgia Tech focuses on developing single particle tracking methods to study the underlying physical principles that govern the dynamics, statistics, mechanics, and self-organization of nanostructured soft materials. Vida is the recipient of the NSF Career Award in 2024 and the ACS PRF Doctoral New Investigator Award in 2023. Congratulations. And the floor is all yours. We're really looking forward to it. Thank you for being here.
Thank you. Thank you. And I'm happy to be part of this webinar today. And what I'm going to share is slightly different from the talks that you've heard, because there is a completely different aspect of using the protochips holder for like a completely different type of work. And I really enjoyed listening to the other talks. I've learned many new things that I think it would be actually very useful for also what we are doing, many new techniques that people are using in the community. And I'm really glad to see that the community is growing and more liquid cell users now we have around the world. So what I'm going to focus on today is on liquid phase TEM for single particle tracking. My group started 2 years ago, so these are a mixture of what I did in my postdoc and what we are currently doing in my group. But what basically is our research is focused on is like is all about single particle tracking, but from hardware, software development, and the application of these single particle tracking methods. So what I'm going to show is mostly, all about gold particles right now, but we are now taking it into next step and using other particles in touch with different environments. And we want to look at the motion of these particles to understand how they interact with their surrounding environment. So with this, I want to start by saying like what excited me to get into liquid phase TEM research was that I was a soft material person. So I was working with filamentous particles, looking at their phase behavior, the thermodynamic phase behavior, and their self-organization. But all we know about this system is either on the macro scale or when we go to the nano scale. It's everything is about ensemble bulk level measurements. So at least that was back in time and when I was doing my PhD. But when we think about these material systems, whether we are thinking about like nanoparticle polymer composites or even like biological soft materials, emulsions, we always have these nanoscale building blocks that they are interacting, and through those interactions, and then they give rise to certain functionalities at larger length scale. So in one case, we want to understand, for example, how to engineer polymer composites, and we want to know how these particles interact with these polymer chains. We have protein molecules in our cells that they either just alone change their conformation in time, so they have some dynamic going on at the nanoscale, or they interact with other protein molecules or lipid membranes, things around them, surrounding environment, that they could give rise to the function that the proteins express at larger length scales, or even like diseases. So understanding of this relationship between the dynamics that goes at the nanoscale and what functionality the systems express at larger length scales requires studying these dynamics directly, because all these systems that I put examples here on this slide are all heterogeneous systems. And when I keep using the word heterogeneous in this talk, and because heterogeneity is something that is like the inherent about these, by looking at, if you look at one point in the system and then move 200 nanometer away and look at another point, the behavior could spatially vary. And sometimes even temporally it could vary. It means that it evolves over time. And so that means you need a direct visualization method to probe them. And just bulk measurements are not good enough for us anymore. So when we talk about direct measurements, then that means we want to have an imaging method that gives us a resolution that has a spatial resolution in comparable to the length scale of our nanoscale building blocks, so something around nanometer. And in terms of temporal dynamics, most of these systems that I talked about have like interesting dynamics going on in the millisecond. And so this is the regime that we're really looking for, 1 nanometer and 1 millisecond resolution. But when we go back to the imaging methods that existed for a long time, for example, optical microscopy is fantastic for looking at these colloidal, microscopic colloidal particles, how they interact, how they self-organize and crystallize. But it is limited by the Abbe diffraction limit. And so that means basically you can't use the visible wavelength light to resolve features smaller than 250 nanometer in size. Of course, there has been a lot of advancements in pushing down this spatial resolution, breaking basically the Abbe diffraction limit, and total internal reflection microscopy, fluorescence microscopy is one of those methods, still limited to 100-ish nanometer. And then we have super resolution microscopy methods, which were awarded the Nobel Prize in chemistry, but they are also limited with 20 nanometer spatial resolution, in addition to the fact that they're not direct real-time visualization techniques. So you need a lot of like post-processing that happens in order to get to that video, but directly just looking at the system, how these nanoscale objects are moving or interacting, that's not possible. And you need to have either objects that are fluorescent, or you need to fluorescently tag them, which itself has its own thing. But and a big issue with that is that you just see a blob of light, so you really can't say if what's the shape of this object. Is it rotationally showing any dynamic? Is it changing its conformation? All those things are basically remaining on, like, you cannot resolve them in optical methods. So what would really work is something that works with the electron beam, because the electron beams now allow us to have spatial resolutions down even below 1 nanometer. But historically we have like cryo-TEM, which I used to use those images, collaborators that were doing cryo-TEM for me in PhD, but you have to vitrify your sample. And most of this system that we initially talked about, them, they have dynamic and kinetics going on that we want to resolve them in time. And dry TEM, normal TEM is also, you have to dry your sample, so that doesn't work. Unfortunately for us now as a community, we have access to liquid phase TEM. And what, of course, there are different forms of liquid phase TEM existing, from the standard holders that now we use, and this is what I'm going to talk about in this talk, but also there is like graphene liquid cells, which are like small pockets. But what if you are like me interested in looking at a single particle tracking method, and you want basically sort of a microfluidic device, then liquid cell holders are the perfect example. And the protochips holder has been a great use for us, when I was a postdoc and now in my group, both using these Poseidon select holders that allow you to flow in and out. And now, of course, different functionalities, changing temperature, monitoring electrochemistry, all in situ, while you can track how these nanoscale objects are moving. So when most of what we're looking at are just nanoparticles that are moving around, and we can change the environment, or we can just, in the first place, understand how they move in the liquid cell in order to establish this technique as a single particle tracking method, which is our goal. So this is the workflow of what we are doing in my group. And we take these silicon nitride E-chips. All of these that I'm showing today are static E-chips, and they have the static spacer, and varying from 50 to 150 nanometer. So these are the two typical sizes that we use because of the size of the particles that we have. And we use gold nanorods of ranging from 20 nanometer in length to 60 nanometer in length. It's mostly aqueous solutions, and at least the videos that I'm showing today. So they are in touch with the silicon nitride window, and mostly we see these particles that are near the membrane. And this is something that I'm sure, like in the community, this is typically what everyone does, because you can keep them in the focal plane, and you can record videos, and you can see how they interact and move, or they're interacting with other objects. Of course, there are a lot of applications in terms of if you are interested in self-assembly of these particles. So most of the time it is close to the membrane. So now we collect these videos, and then there are like terabytes of data, and then we process them into track by tracking the position and orientation of these particles over time, and we arrive at these spatiotemporal trajectories that you see here. And that's telling you how these particles are moving in time, how they change their orientation in time, and so on and so forth. And this motion, I'm going to say it in the beginning, is definitely impacted by the interaction that they have with the environment. And the entire goal in this project that I'm going to talk about is like what sort of interactions exist and how those interactions give rise to the type of motions that we see. Because as the community now, we know that none of these particles in the liquid phase TEM are moving the way that if you would have observed them in a normal optical microscope would move. So the question is why, and this is what we are trying to answer here. So part of my research, both as postdoc and now in my group, we have been focusing on this question of how does the electron beam affect the motion of nanoparticles in liquid phase TEM. And this is something that was mentioned and also in the both of the previous talks. In different communities and different research areas, it has different impact. For us, it means that if we want to use it as a single particle tracking method, we need to understand as a baseline how particles move in the presence of electron beam, because the electron beam is there. It is the tool that allows us to visualize all these nanoscale dynamics. And so we can't get rid of it, but we need to understand it. And once we have that baseline understanding, then if we change the environment to anything, or it's changing the membrane, or changing the type of the particle, then we know what we should expect, basically. So for this type of study, what we have done is that we have looked at the range of particle length and in a range of electron beam dose rates on these static chips in silicon nitride E-chips, and observing their motion, recording movies, and then analyzing these movies in order to understand this type of mechanism of motion. And so then on the next step of that is, of course, when you have that understanding and you have that control, is there a way to exploit that electron beam for engineering? And this is something that I heard other people are also doing in the community now. So when you're purposefully making a bubble and trying to increase your spatial resolution, you're actually using that fact that the bubble is generated by the electron beam. So there are multiple aspects that you can use this, but for us, it is there a way that I'm going to bring an example of how you can do in situ liftoff using the electron beam. And this is one of the examples of the work that we did when I was in Paul Avisada's group at Berkeley. And I'm going to go over that briefly, but of course, that's mostly Hodak's work, one of the first PhD students working with one of the first generation of these holders. And this is how I got into also the liquid phase TEM research. So now, if I show you two example videos from liquid phase TEM, the left-hand side, you see a particle collected at an electron beam dose rate of 15 electron per angstrom square second, and on the right-hand side, you see the dose rate of 35 electron per angstrom square second. What you notice, obviously, is that if I'm from left to right, on the left-hand side, we have these particles that the particles moving very limited, and the distances, the steps that it takes is very limited. It's confined to its original position. But on the right-hand side, the particle moves not only faster, but also it takes larger step sizes and it diffuses more. So if I now track the position of this particle over time and plot it for this during the same amount of time and scale it to the same size. So even though I'm talking about dose rate here, you can think about it that cumulative dose is also the same in both of these cases. What you see is that there is a trend, and that trend is that for sure the diffusion coefficient changes as you increase this electron beam dose rate or cumulative dose, however we want to think about it. So so of course the diffusion coefficient changes, and this diffusion coefficient is still, even on the right hand side plot, is much slower than what you would get on a turf or on any type of optical any fluorescence microscopy. It's about almost 6 orders of magnitude smaller. And this is something that has been verified over and over. Everyone, different universities using liquid cell holders, they always observe the same thing. But now if I magnify these trajectories for you, you'll see that it's not just the diffusion coefficient that is different in those two cases, but also the pattern of the diffusion is completely different. So on the left-hand side, you see these particles that have very clustered points, and there are two basically main clusters that you see, but on the left-hand side, and on the right-hand side, you see these small clusters, but there's a lot of jumps. So it is the diffusion, when I say the pattern, it basically tells you that the underlying diffusion mechanism is also changing with the dose rate and the cumulative dose. And so the question becomes, what is going on with the electron beam in the system? What does it do, and how does it change the diffusion mechanism? And I mean, visually there is some change, but what is this change and what does it tell us about the environment? So this was how my basically the first project working with liquid phase TEM, trying to understand this. And when I started on this, I thought about, okay, I'm going to collect systematically these trajectories. So I'm going to keep the time the same, and I'm going to change the dose rate, and I'm going to look at how particle diffusion coefficient changes. And then I can also look at if they left them in their entirety, like if they diffuse, I can look at and magnify it and look at how the diffusion pattern is evolving over by increasing the dose rate. So there's obviously systematically there is a change if I collected multiple of these trajectories at different dose rates from 2 to 49 electron per angstrom square second. And this was something that you would clearly see without a doubt every time that you do this experiment. And interestingly, to some level, this is reversible. So if I decrease the dose rate a little bit, it goes back to what it was, so lower diffusion. So it's not something that you would say it would permanently change the chamber environment. So then what we did was that, okay, so now there is a challenge in analyzing these data that we get. So there's a lot of, you will see a lot of artificial intelligence, how it gets integrated with these liquid phase TEM research. But the reason for using it is that the diffusion is so complex that there is no model existing out there that I can tell you that this is exactly this type of diffusion. And we're going to get to that. But what initially we could do was that to say like roughly at high dose rate and low dose rate, what is it similar to? And there are some ideal diffusion processes out there that are described for different scenarios. And one of them is Brownian motion, which all of us are familiar with. If there was no electron beam, I would have expected to see these particles moving in a Brownian beam. But then there is fractional Brownian motion, which is a subcategory of Brownian motion, but it is usually happening when you have a particle moving in a viscoelastic network. It's like a gel-like system. And then you have continuous time random walk, which is a trapping and hopping event that has been visually, many people have reported it, but it is basically the phenomenon, the mechanism governing that is called continuous time random walk. Very, very different phenomenon mechanism from the other two. So both fractional Brownian motion and continuous time random walk, which I call them FBM and CTRW, are type of anomalous or subdiffusive motion. And so what we did was that I developed this neural network we called MONET, it's or MotionNet. And MONET is simply a convolutional dilated neural network that is trained on thousands of trajectories from these ideal diffusion classes. So something that you can simulate. And then I analyzed a lot of videos from these single particle gold nanorods moving in the liquid phase TEM and near the silicon nitride membrane in water. And then we get the X and Y coordinate of the particle over time. And then if you input it now into this neural network, then it would output for you the probability of having each of these classes of diffusion. So with what probability you have fractional Brownian motion, with what probability you have Brownian and CTRW. And we chose this trajectory length that we analyze as 300 frame point because sometimes you have very short trajectories. Sometimes you actually want to segment your trajectory into short pieces so that you can swipe the window all through your trajectory to see if there's a time evolution in the behavior of a particle in liquid cell. So that allowed us to do that. And at the same time, it was long enough that you would have accuracy that you need. So on ideal processes, it has about 90% accuracy. And it is not just applicable to liquid phase TEM, by the way, it is applicable to any type of microscopy trajectory data. So what we did then after that was that we applied it on 460 mini trajectories that are each 300 frame long from 40 nanoparticles in about like 5-6 different experiments that we collected these in dose rates from 2 to 49. So in total, you're right now looking at 40 pie charts, each representing a particle behavior, average behavior for a particle at that dose rate. And then what obviously is very clear is that as I increase the electron beam dose rates from 2 to 49, there is this crossover from fractional Brownian motion shown in green to continuous time random walk shown in blue. But okay, so there is obviously a crossover. We validated what we saw initially in the video, and we thought that there is something in the diffusion mechanism changing. So at least probability-wise, it's more closer to continuous time random walk at high dose rate and more close to fractional Brownian motion at low dose rate. But what does this tell us about the environment of the liquid cell once you increase the electron beam dose rate? Well, if you think about the fractional Brownian motion, again, it is related to a particle moving in a viscoelastic environment, like a gel-like environment. And continuous time random walk is when you have a heterogeneous environment where you have these potential wells or interaction sites that a particle interacts with, gets trapped, and then it detraps and make a jump to another spot. on that heterogeneous membrane. So now if we put this into context of our experimental setup, what we hypothesized at the time, and this is very something that, so if you think about the silicon nitride membrane, it has silanol group on it. And this is something that we are not the first people who observed it. Of course, like people like Otkor Mirzaidov or Taylor Wall using silicon nitride membranes also, and many others. I'm of course forgetting names, but many people in the liquid phase TEM community reported before us that there are silanol groups on the surface, especially when you do easy glow or plasma cleaning. You want to make your surface hydrophilic. And so that, of course, the water wets the surface better, you have a better assembly, but as a result of that, you are making the surface negatively charged. So there are works by Mirzaidov that shows that if you change the type of surface treatment, like chemical treatments that you have on the surface, you get different amounts of or extent of interaction with the surface. But here the particles are fixed. They have all coated. They're all coated with CTAB or CTAC ligand, which is positively charged, and the surface has silanol. So there is, of course, the interactions between the particle and the membrane, and that's why they remain in the focal plane during the imaging time. But what we hypothesized at the time, what would be happening is that at low dose rate, you have these particles interacting with silanol groups, so they're trapped, and that's why I'm showing them these potential wells that you can see. So there is an energy potential well with some energy well depth, and the particle is in there. But within that potential well, within that interaction site with a bunch of silanol molecular groups, probably there is some viscoelasticity. And so we went back to the literature. So there is a lot of work, and this is still an ongoing research problem, that if you have confined geometries, water in touch with graphene or silicon nitride, does the thermodynamic properties of water change? And indeed it does. And so the question was that there were works back in 60s that claimed that if you have water next to the silanol group, and it's a confined environment, you could, within few nanometers of the surface, you can make these hydrogen bonded layers that then represent some viscoelastic effects. So the interactions that the particle has along with this increased elasticity and viscoelastic basically behavior near the surface could show up as a type of motion that we see as fractional Brownian motion that typically outside of this liquid cell were associated with particles diffusing, I don't know, in polymer gels or things that are more viscoelastic. So that was like, okay, so if this is something that people studied before near borosilicate surfaces, or in general like surfaces that have silanol, it is consistent what we see. But what happens when you increase the dose rate? We all know, this last slide was talking about it, and that if we increase the electron beam dose rate, then the radiolysis happens, the pH of the environment change, and there is like a lot of work by Francis Ross group, Schneider, and many others looking at the pH of the environment. And so the question is that when you increase that electron beam dose rate and cumulative ox of the electron beam, and then you have these protons hanging out in the solution, then they can passivate. Can they basically passivate these silanol groups? And if that's the case, then the energy barrier of these potential wells are lower, and so the particle should be able, with the same thermal energy, should be able to now overcome this potential well and jump to another potential well and another one, which now resembles a heterogeneous, moving over a heterogeneous landscape. And there is no reason for us to think that the temperature is changing here. I mean, with the new generation of holders, now we can look at monitor the bulk temperature in the solution. And in general, I don't think this is something that is accepted in the community, that temperature doesn't really change. At least in the solution level, that, I mean, I'm not talking about locally around like maybe few angstrom around the particle, because that depends maybe on the type of material that particle is made of. But for the solution within this field of view that we're looking at, there's no way the temperature would locally change. But what is changing is these interactions with the particle, and that is how the electron beam or the radiolysis species can change those type of interactions, and that allows us to observe more dynamic particles that diffuse more, and particles that detrap and escape and jump. So this seemed at a time very consistent with what we were observing, but there was one piece of information as I was finishing on my postdoc. I was like, but there is, when we look at this data, there's one thing that this still makes me wonder, and that what type of a crossover we are talking about here. There is, when you think about the middle range dose rate, for example, the dose rate of 15 or 20 electron per angstrom square second, then you see these particles that show both of the characteristics. They show both these regions that are confined, and they showed some long jumps. And when you ask our Mones neural network software to classify the behavior in this trajectory, overall for this particle, full trajectory that you're looking at, it is almost like 75% fractional Brownian motion and some good probability among like 20% maybe, or it's slightly less than that, continuous time random walk. And I would ignore the Brownian part because Brownian and a fractional Brownian are very similar, and there could be an error in identifying the neural network identifying these two, distinguishing them from each other because they come from the same family almost. But continuous time random walk, when it says it has continuous time random walk, it really has those features, and visually we can see those big step jumps. So then we started to think that, okay, so now this is a good use of our software because we can scan 300 by 300 pieces and then start to look at how does the behavior evolve over the trajectory over time. And so we can note, what you can actually see in the middle figure is that the probability of continuous time random walk, the mechanism that shows up as big jumps, spikes up when you have the jump, and the probability of fractional Brownian motion will spike up when you have this cluster point. And so really both of these mechanisms coexist. And so we started to think about that at that time, that is there the coexistence of these two. And so you are just, when you're doing collection of your videos from the particles in the liquid cell, you only get to see one window at a time. And and what I mean by that is that if you're looking at low dose sample, you're collecting your videos at low dose rates, if you could have waited for hours, then you would have seen your particle making a big jump. And when you're working at high dose rates, if you had a camera that would allow you to capture videos at nanoseconds, then maybe you would have seen that confined type of motion. So both FBM and CTRW coexisting. And so when this was something on my mind, and when we started at Georgia Tech, my first student collected a lot of data. So you're looking at a lot of particle trajectories here. And what we did was that we used this, so let's say the red data points here are showing the liquid phase TEM data. And then now when we encode them into a low dimensional space, so this is a TSNE test, T-distributed stochastic neighbor embedding. So it just shows you what is the low dimensional. If I just cast everything into a dimension of 2, where are the liquid phase TEM data sitting? And it's everywhere. And then on the blue data points are showing you continuous time random walk processes of different values of alpha, and the alpha just tells you how subdiffusive a trajectory is, with alpha 0.1 very subdiffusive and alpha 1 almost Brownian motion. And the same is for green data points that are fractional Brownian motion trajectories going from 0.1 to 1. And liquid phase TEM data is everywhere, so it's covering both fractional Brownian motion at different levels of anomaly as well as continuous time random walk. So it was a sort of a hand baby verification of that liquid phase TEM data has all of these features, and it is very complex, and indeed there is no closed form model that would capture what we see in this experiments. And so and for many of these experiments that we do in future, or many of the tasks that we want to do in future, we need models that would describe this motion so that we can infer interactions from them, we can generate trajectories, so we could just simulate trajectories resembling liquid cell environment. And that wasn't there. Obviously there is no a stochastic model or Langevin based model that we could use for that. So what we started to do was that then we developed an unsupervised model, which is a generative model that would take in liquid phase TEM data and then generate and try to reconstruct it, and not just reconstruct every snapshot, but reconstructing all the characteristics of the liquid phase TEM data, including the distribution of displacement of the particles, or like the anti-correlation that exists between the steps of a trajectory. And it does this by encoding trajectories into a low dimensional space that we call it a latent space. It's a probabilistic type of model, so that it is captured, each latent variable is captured by a mean value mu and a variance sigma, and then it tries to reconstruct it. And through this process, of course, there's like these layers that have the attention mechanism similar to what is like in ChatGPT, which try to learn the relationship between the steps that a particle take. And it is informed, it has a loss function that is informed by the physics of the trajectory. So what do we know about characteristics of liquid phase TEM data and how those relates to physical features, and how can we help the neural network reconstruct those better? And so when we did this, then we could start to analyze, okay, what does the neural network, obviously it was able to learn it, but what does it learn about the data? Because it should be able to boil it down into all these latent space features, and those are the features of liquid phase TEM data. So when we did those type of analysis, we could look at this gray latent space, different spots have different sort of, give you different sort of trajectories with low dose characteristics or trajectories with high dose characteristics. And so we did analysis, a lot of a statistical analysis on those latent space, and there are 12 latent space in total, and latent variables total in our latent space, and 2 of those seems to be capturing 2 significant property of our liquid phase TEM data. And one, and these 2 characteristics, as you can see here, the matrix on the bottom is showing the yellow and blue color. So the blue means that there is a high correlation. So this is a correlation matrix. The latent variable 12 is very much correlated with alpha of fractional Brownian motion, which means that how the characteristics of fractional Brownian motion trajectories, which is like viscoelasticity and all those features that we saw at low dose rate, and latent variable 2 is then correlated with continuous time random walk anomalous characteristics, which is making those jumps and being showing a distribution of the displacement that is very non-Gaussian, which is very far from Brownian and fractional Brownian motion family. So by doing this analysis, then we were able to say, okay, now we have knobs that we can change, and we can generate trajectories from liquid phase TEM data without doing the experiment and saying, okay, can I generate low dose trajectories? Can I generate high dose trajectories? Can I mix them at different levels? And so these are just like we have a simulation box, a black box model that can simulate trajectories just like liquid phase TEM data. And these, of course, we can tune their value between -3 and +3, which are the standard deviation of these, -3 and +3 standard deviation of those latent variables. So now, once we have this, one reason particularly very interested in is that if you want to, if you think about future of automating electron microscopes and you want to generate synthetic trajectories and put that into a loop of microscopy so that you don't need to do all those experiments over and over, then you can just sample from different regions of this latent space. As you can see here, these red points that we're sampling from the latent space, you can actually change the behavior of the trajectory or in time, and that like in real time you can see how it would behave. And this is exactly like if you think about, if I change my dose rate during the experiment, how does the trajectory supposed to evolve in time as I'm watching this experiment? And so you have access to a lot of trajectories from just a black box simulator in that way. And then so this is, of course, one part of the story that we want to understand. We want to be able to model the liquid phase TEM trajectories. But the last piece that I want to talk about is very quickly is that, is there a way now for us to use the electron beam? Because we know what it does. We know it does the radiolysis, but can we use that fact that there's these species generated? This was, I think this is a very elegant work that I was had the pleasure of working with Horak on this work, and I always advertise it because it's very beautiful. You can think about this silicon nitride membrane and then and chips, and then taking the chips to the nanolithography room and try to lithograph these nanorods on the surface. Now you can choose to have your rods. These are lithograph rods, but they have a base of chromium or titanium that you, when you lithograph them on the surface. What we know, based on all the experience with working with the liquid cell and different materials in the liquid cell, is that chromium etches away very easily with the radiolysis species that are generated, including hydroxyl radicals, but titanium remains intact. So what we could do is that we leverage this fact. So we make an array of rods on the surface, some attached with chromium, some attached with titanium, depending on how we want to play with this array of particles. And then once we add water, and then we assemble the chip. These are again static chips, and then we look at them under electron beam and shine the electron beam. Then the radiolysis starts to happen. These are dose rates. I believe these are above 50. Dose rates above 50 that you actually start to really. It might be even 100. I don't remember the number exactly, but this is very well beyond that point that you actually intentionally make a lot of radiolysis species. And so the chromium starts to etch away, and those rods that were attached with chromium are lifted off the surface, and they start to now move within this array of fixed nanorods. And then you can watch them, how do they assemble. So here's a simple example of like if you have these 3x3 array, and the middle rod is just attached with chromium, so it's lifted off, and then it starts to interact within this fixed array. And this allows us to model the interaction of these rods with this array of fixed arrays. So you make a synthetic energy landscape, and then you watch how these particles interact, and then you can back calculate parameters like the bias screening length, or the how much was the charge density of the particle. And so this by just knowing these parameters, then you can go back to the lithography room and then put the particles in different spacings. And then you can see that you can get different type of assembly. You can get a zigzag assembly, or you can get a head-to-head assembly. The parameters are fixed, depending, we know how much ligand now we have on the particle. We could indirectly characterize it through the physical parameters, including the bias screening length again, but just the spacing between the particles, you could use it as a knob to get different form of self-assembly of these particles. And I think this is a form of like thinking about, well, we can use the electron beam actually as a tool. And if we are planning to move these rods to make nanoelectronic devices or things like structures like that, then this is a tool. It's a method of doing in situ liftoff, basically using these nanorods. And there are, of course, more exciting work hopefully coming out of our group soon in terms of how we can use the electron beam. I hope that I can share this all with you very soon. We just started very brand new, so hopefully more work are going to be out soon to talk about them. But partnerships holders have been a great help for us. Of course, none of this would have been possible without the support of my group. I mean, all I talk about is now I used to do experiments, but now these are actually the people who do the experiments. I'm very fortunate to work with them. And mostly what I showed here today is the work from Zane, my first student. And yes, and I want to thank all the prototypes for this webinar, organizing this webinar, meeting everyone and hearing about the exciting work in the community. And also from all of you, thank you all for joining, and thank all the funding agencies for supporting our work. And I'm happy to answer your questions.
Wonderful. Vida, thank you so much. What an awesome presentation. And you're a very engaging speaker. So thank you so much.
Thank you.
I do have a few questions here that were submitted for you. So let's go through. The first one says, thank you for a nice presentation. You're doing a lot of diffusion work. What has your observation been related to the suppressed or slower than expected Brownian motion that's been reported? And is that a benefit or hindrance to your studies?
If I was going to say that I would have wished there would be Brownian, obviously, because it would have made my life much easier. I could have just changed the environment and then do my single particle tracking at a resolution that was unbeatable with a spatial and temporal resolution that liquid cell has, is basically unbeatable by any other microscopy technique. So we don't have Brownian. And it's not just that we don't have Brownian, or it's not even a slowed Brownian. So it is a non-Brownian that we are trying to learn it, and we are trying to classify it. So all our work has been focused on that. It is a challenge, but I think it is doable. And that's basically the motivation that we have in our group, that we are going to use it as a baseline if we understand how things work at different dose rates, at different particle size, with different particle materials. Then as you can imagine, if I change now the environment from, I don't know, water to glycerol, then I should be able to exactly tell you how the particle diffusion would change, because I have a baseline understanding of how it works in water. So now I'm just changing the solvent to something that's more viscous. So it is very different. I wish it would have been the same as optical, but it is what it is. And this is the only tool that we have that gives us this resolution. So that's our hope, that we are developing it as a single particle tracking tool.
Very nice. Very nice. Thank you. The next question I have is, do you see any differences in motion based on liquid thickness? So with the liquid cells, you can image in the corner versus the center. How do you approach that?
They're the same because you're looking at everything near the window again. If it was in the middle, like it could be in the corner of the edge of the cell, as you said, or it could be in the middle, but all the time it is near the window. If it was in the somewhere in middle of the chamber, I mean in the Z height, it was in the middle of the chamber, then it would have been moving so fast because there's less of interaction. At least that's what we're thinking, is that those particles, we don't even see them within the field of view that we get in the TEM, and they move super fast. So hopefully there exist maybe some Brownian ones out there that we don't have yet the tools to probe them. But everything remains in focal plane. That means they're interacting, and that means they're next to the window. And that at that point, it doesn't matter where you're capturing. We have all of our data that we're looking at. It's a combination of particles in the corner and particles in middle of the cell, and they all show the same behavior.
Very interesting. Good, good. The next question that is listed here, thank you for the presentation, very nice. You mentioned, they didn't put a slide here, but if you mentioned that when under low dose rates, if you observe them for multiple hours, you might see them do the big jumps of high dose rates. Do you think there's an effect of cumulative dose as well, as opposed to only dose rates then?
So we have been looking at both. Initially, when I started working with liquid cells, I was mostly thinking about dose rate. And then I started gradually thinking about, but what about the cumulative dose? I would say pretty much it's consistent, because like some of the figures early on I showed them during the same amount of acquisition time, and both the total cumulative dose and the dose rate were changing, and so the trend was consistent. So it could be that it was convoluted. We have some ongoing studies right now with the cumulative dose. Both of them basically, it's as if you think about it, even in the case of cumulative, it is the same effect as the dose rate. So more electrons interacting with the system and more radiolysis species generated. So the effect is the same, but it's just a different way of categorization. It's not too different in terms of what you get in the end, but keep that in mind that these processes are reversible. The diffusion of ions are much faster than the processes that we are probing. And so if you go back to the particle at lower, if you decrease your dose rate, you would see the particle slow down again. And this is an interesting thing about liquid cell.
Yeah, very nice. And then I just have a question for you, just out of my own curiosity. How are you going about measuring dose? Do you have a Faraday cup in your TEM, or do you use like the screens, or just like how do you measure it?
So we don't have the dose. We don't have currently a Faraday cup holder. So what we do is that we have the conversion factor of the camera from Gaton. We use a one view camera, and then we have an in-house code that calculates how much electrons reach the screen, and based on that, calculating the dose that we have. So the dose that I say might not be exactly the dose that you measure with a TM that you're working on with, but the ratio of the dose rate in the experiments that I do is the same. Like,
you got that relative.
You can make sure that next time that, qualitatively, the next time that you do the experiment, you exactly get the same dose rate as your last time, because the camera is the same, everything is the same. But it's not the same with another lab, I would say.
I gotcha. Okay, that's everything that had come in through the questions. Thank you so much for a really lovely talk. It was really, really interesting. And thank you so much for being here in general. We really appreciate you. I know CMOS appreciates all of our speakers today, Sanjay, Ahmed, Vida, for coming in. I know there's a lot of interest at CMOS too for the technique. And we'll be able to share these presentations too with anybody interested in this technique in general. And so the work you're doing, kind of forging this cutting edge research is really important to the community, and there's a huge interest in it as well. So I know we'll be able to share your talks across the world for people who are interested in kind of breaking into this area, but you guys are really leading it. So nice job. And again, thank you so much for being here.
Thank you.
Yeah.
again.
Thanks, everybody. And everybody who joined, thank you so much. If you missed anything, we'll record, it's been recorded and we'll share it afterwards. We'll post it up on some public web platforms for you to view. Otherwise, I hope everyone has a good afternoon, and I'm sure we'll see you again in the future sometime soon. Thank you. Bye,
Thank
everybody.
you all. Bye bye.